In biotechnology, DNA extraction is a method that separates a targeted DNA from an organism. Purification is an essential procedure in this method, and a spectrophotometer is an instrument used to detect the purity level of the DNA by referring to the wavelength displayed. In this experiment, onions were diced and divided into two portions, where one of them is mashed. These onions are then transferred into a beaker containing lysis solution and will be filtered before extracting the DNA. There are three objectives, which is to learn the proper way to use a spectrophotometer, to observe the differences between DNA extracted from mashed onion and chopped onion, and lastly to analyze the level of purity of the DNA extracted.
Conical flask and filter funnel, filter paper, measuring cylinder, test tube, spatula pasta and mortar, micropipette, knife and sample onion, beaker, tea buffer, isopropanol, IC solution, microcentrifuge tube, bench top scale, spectrophotometer, microscope.
First, an onion was chopped into 1 cm cube and divided into two portions, labeled as P1 and P2. Both of these portions are weighed using an analytical balance respectively. Ensure that both P1 and P2 are 5 gram. By using a pestle and a mortar, the P2 onion was mashed until it becomes a paste-like texture.
The chopped P1 and mashed P2 onion portions were transferred into separate beakers containing 25 ml of onion lacy solution, thoroughly mixed. The beakers were placed in a water bath set to 60 Celsius for 10 minutes.
Next, the beakers were removed from the water bath and immediately placed into an ice bath. Both were swirled gently until the contents felt cold to the touch. Then, the mixture was filtered to remove cell debris by pouring the contents through Wetman filter paper number 1 into clear.
The 10 ml of the filtrate from each beaker P1 and P2 is transferred into separate test tube with labeling as P1 where is chopped onion and P2 is mashed onion. Then the 10 ml of isopropanol is added slowly into each test tube and is mixed gently until white stringy DNA precipitates out of the solution. Next, the differences between the sample P1 and P2 regarding DNA yield and appearance is observed.
The DNA was collected by gently spooling it onto a glass rod or a wooden stick. The DNA was examined under the microscope. Then the collected DNA was transferred into a TE buffer. Lastly, the purity of each DNA sample is determined by using spectrophotometer.
Now let's talk about the result that we obtained. First, start off with the P1 sample, which is the chopped onion. By using the information that we have, substitute them into the formula of concentration DNA, which is 50 micrograms per milliliter times with absorbance at 260 times with the dilution factor. We get 16,610 micrograms per milliliter. For DNA purity, which is absorbance at 260 over absorbance over 280, we got 1.010, which is not pure. Moving on to P2 sample, which is the mashed onion. By using the information that we have, substitute into the concentration DNA formula, which is 50 micrograms per milliliter times with absorbance at 260 times with the dilution factor, we get 16,680 micrograms per milliliter. Now to find the DNA purity, absorbance at 260 over absorbance at 280, we get 1.430. 1.430 is not in the range between 1.8 to 1.9, which is not pure.
Here are the functions of all the chemical solution that is used in this experiment. Next one is the explanation for the difference in observation between mashed onion and chopped onion. The difference in observation between mashed onion and chopped onion are caused by the surface area of the onions, where mashed onion have bigger surface area, whereas chopped onion have smaller surface area. Thus, the rate of absorption of the lysis buffer solution into the cells is different, where mashed onion have higher rate of absorption than chopped onion. When isopropanol is added, the mashed onion produces a DNA-rich solution, allowing a visible white stringy to form. On the other hand, the chopped onion does not form a visible white stringy, or if it forms, it's only a short one, as the DNA concentration is lower. Both sample is not pure, as there may be a contamination on the sample, or the onion cells is not completely lysis in the solution, causing the DNA to be still intact with the cell membrane. Therefore, precaution steps could be taken to minimize any error during the experiment, where the spectrophotometer is cleaned with alcohol before use.
With that being said, we can conclude that the plant tissue can be isolated by a breakdown of cell wall or membrane to release the DNA using a lysis solution to remove proteins that are bound to the DNA. The better DNA extraction method by using spectrophotometer is mesh the samples because this increases the cell surface, efficient cell lysis, better homogenization, and minimize DNA sharing compared to chop the samples.